Good afternoon, everyone. My name is Riya Patel. I'll be presenting on non-enzymatic detection of oxalic acid in fungi, S.sclerotiorum infected theropods. Um, I'm presenting it on behalf of my team as they could not make it. So plant pathogens, what are they? Pathogens are any disease-causing microbes, can be a bacteria, virus, fungi. And why is it very particular to detect pathogens in plants is because diseases in plants, if they are prevalent, can lead to huge economic losses. And about 20 to 30% of the economical loss in agricultural sector has been known because of diseases which occur due to pests as well as due to microbes. Early detection will be a boon because the traditional methods of detection, which are commonly used even in agricultural settings, are like microscopic visualization, PCR, ELISA. All of these are laboratory techniques which require skilled labor and also sophisticated instruments, and therefore are also time consuming. So having certain other techniques or a biosensing platform developed in an agricultural setting would serve as an advantage to overcome the preceding technologies. Infection spread widely across different plantations can occur, as I mentioned, due to disease-causing bacterial or fungal agents. And the biggest issue is any contaminated sample the disease can easily spread via irrigation or due to airborne infections. Uh, Sclerotiorum is a highly destructive plant fungus. It is known to cause the cotton root or the white mold, as the disease is called. And it has a wide host range of economically important crops. What I would be discussing is the application of a sensor to detect this fungus in peas plant. This spreads via airborne ascospores, or it can also spread in soil if the fungus is soil-borne and also during irrigation process. Another disadvantage of having this fungus in the plant sector is that it is very difficult to control the spread of this disease, and it also has seen that there are a lot of varieties of being resistant to even any kind of pesticides or control agents. Electrochemical detection of pathogenicity. So there are various ways where in electrochemical methods have been developed and utilized in biosensing methods. Um, Another indirect method if to detect, which can be label free, is to detect the presence of oxalic acid in a host cell during the infection. The amount of oxalic acid which is secreted by the infected host plant directly corresponds to the fungal pathogen intensity. Quantitative estimation of oxalic acid can be an indirect method to assess the disease condition in the plant. It can serve as a cost-effective, simple, rapid, and yet a very sensitive electrochemical method if we can detect oxalic acid content in an infected plant sample. Um, Screen printed electrodes are serving as an ideal sensor platform for agricultural sensing application. The advantages of using screen printed electrodes is there's an ease of fabrication, and you can also reproduce it at a faster pace. Small size electrodes are possible to be printed, and we can also have disposable electrodes, making it very user friendly. The device which was utilized for this work is a three terminal electrode working reference and counter electrode, where the working and the counter electrodes was just a carbon paste, while the reference electrode utilized was a AgAgCl paste. They were screen printed as shown in the fabrication steps in the image. First step is doing the electrochemical characterization of the screen printed electrode and then confirming the functionality of the electrode. So the functionality of the screen printed electrode was confirmed by performing cyclic voltammetry measurements using a standard redox solution of potassium ferricyanide with KCl as a supporting electrolyte. Both the oxidation as well as the reduction peak current increased with an increasing scan rate, and the linear behavior of the magnitude of peak currents with a square root scan is seen, indicating the reaction is diffusion governed. How is the non-enzymatic detection of oxalic acid was carried out? We utilized a paper-based SPE for quantitative detection of the electroactive pathogen marker, which is oxalic acid in real infected samples. It was a non-enzymatic approach, looked for the electrochemical oxidation of oxalic acid and measured it electrochemically. The redox reaction of oxalic acid happens at the working electrode, and the current quantifies the amount of oxalic acid. As mentioned earlier, the amount of oxalic acid would correspond indirectly to the effect of the disease in the plant. First step was bringing the calibration curve for the oxalic acid to know different concentration limit for the developed sensor. Redox reaction occurs and happens at the working electrode, and peak current quantifies the amount of oxalic acid in the sample. Calibration curve was plotted, and this was used as a standard reference for testing with the applied samples. Detection of oxalic acid was carried out in infected pear pods which were locally obtained, first based on the visual infection, and then bringing about a very small water extraction protocol without any chemical additives was utilized for not isolating, but extracting the oxalic acid from this infected pear pods. These samples were then characterized using the current measurement. Electrochemical measurements were done for the extracted solution with 0.1 molar ammonium sulfate as a supporting electrolyte. And using the curve, which was plotted with the standard concentration of oxalic acid, the amount in the test samples was determined. Concentration of oxalic acid obtained for the least infected sample was about 43 micromolar, while it increased to 438 micromolar for severely infected samples. The least infected and severely infected is based upon the visual examination of the samples which were obtained. Expected outcome is among the various electrochemical methods reported, quantitative determination of oxalic acid in real infected pear pods was demonstrated. The results can be used to correlate that oxalic acid production increases with the intensity of the infection in affected plants. So this served as an indirect measurement of knowing the intensity of the infection in plants by measuring or knowing the concentration of oxalic acid. The simple and rapid electrode fabrication, technical simplicity, and feasibility for real sample analysis were the key features for this electrochemical sensor. This served as one of the preliminary works and an approach for plant health diagnostic sensor development. And we're looking forward for also analyzing various other parameters along with oxalic acid. Yeah, thank you.